Narcolepsy type 1, or NT1, is characterized by a precise biological deletion. It is the result of the autoimmune destruction of exactly 70,000 to 90,000 orexin-producing neurons located deep within the hypothalamus. These specific neurons manufacture orexin, a neuropeptide that functions as a critical stabilizing signal. It projects across the brainstem and diencephalon, constantly enforcing the boundaries between sleep and wakefulness, so the system does not oscillate unpredictably between the two states. This chart shows the physical reality of that missing neuropeptide. A healthy individual maintains cerebrospinal fluid, or CSF, orexin levels above 200 picograms per milliliter. The strict diagnostic threshold for NT1 requires that level to fall below 110 picograms per milliliter. This targeted neuronal destruction is strongly linked to a specific genetic marker, the human leukocyte antigen variant known as HLA-DQB1*0602. But carrying this gene is not enough. The immune system requires a specific environmental trigger to activate the destruction of the orexin network. The medical community observed clear proof of this trigger mechanism following the 2009 to 2010 H1N1 influenza pandemic. Across multiple European countries, an acute spike in NT1 cases occurred among children and adolescents immediately following specific vaccination campaigns. Epidemiological studies traced this outbreak to Pandemrix, an AS03 adjuvanted H1N1 vaccine. The immune response generated by the vaccine cross-reacted with the exact orexin-producing neurons in genetically susceptible individuals, actively destroying them. NT1 is a highly localized neurodegenerative state with a perfectly understood molecular cause. The physical machinery that produces orexin has been deleted by the immune system. The standard medical response to this known, permanent deficit relies entirely on lifelong, daily symptomatic polypharmacy. Patients are prescribed heavy chemical stimulants and sedatives to manually suppress the downstream symptoms of sleepiness and cataplexy, while the underlying biological void is left completely unaddressed. The most effective medication available for preventing physical collapse, sodium oxybate, is a highly restricted central nervous system depressant. It requires twice-nightly dosing, meaning patients must wake up in the middle of the night to take their second dose, and exacts a massive financial toll of tens of thousands of pounds every single year. Since the year 2000, researchers have known the exact molecular deficit and the genetic architecture of narcolepsy. Yet, over 2 decades later, no curative therapy has advanced to clinical translation. A complete open-source technical blueprint for a permanent genetic cure can be assembled today. We can map the entire process using existing, off-the-shelf biotechnology. Curing narcolepsy is no longer a biological mystery requiring a scientific breakthrough. It's a straightforward engineering problem requiring the correct assembly of validated parts. The primary mechanical hurdle for this cure is obvious. We need to replace a specific neuropeptide, but the original 90,000 hypothalamic neurons that act as the manufacturing plant have been permanently destroyed. Preclinical models solve this through the ectopic principle. Research demonstrates that precise anatomical replacement is unnecessary. If you can force any living neuron in the central nervous system to produce and release orexin, the brain's receptors will absorb it, and normal sleep-wake phenotypes are restored. To achieve this, we need a microscopic delivery vehicle capable of navigating the central nervous system. The optimal tool for this is adeno-associated virus serotype 9, or AAV9. AAV9 is a replication-incompetent viral vector. Its natural reproductive genes are hollowed out, leaving an empty structural shell designed to carry custom genetic material. We select the AAV9 serotype specifically for its unique mechanical advantage. Once introduced to the cerebrospinal fluid, it possesses a superior ability to cross the pia-glial barrier and distribute its payload deeply into the brain parenchyma. This specific vector has an extensive human safety record. AAV9 is the biological foundation for widely approved, globally distributed pediatric gene therapies, such as Zolgensma. AAV9 provides the necessary replication-incompetent transport vehicle to bypass all of the brain's defensive barriers and access the required neural network. Inside the empty AAV9 shell, we insert the human synapsin-1, or HSyn, promoter between the viral ITRs. It restricts activity exclusively to neurons. Directly downstream, we introduce the core therapeutic payload, the human pre-pro-orexin coding sequence, known as HCRT. We utilize the full precursor sequence rather than just the mature peptides. This forces the newly transduced neurons to use their internal secretory machinery to process and naturally co-release both orexin A and orexin B. Utilizing the exact native human amino acid sequence strictly minimizes any theoretical risk of the immune system entirely rejecting the protein, or of the peptide failing to bind correctly to the brain's receptors. This architecture guarantees that only the correct physiological environment, living neurons, will manufacture and regulate the missing neuropeptides. To maximize expression, we add two final regulatory elements, the WPRE and the BGH poly-A signal. The WPRE enhances messenger RNA stability, multiplying orexin production by a factor of 2 to 8. Tallying these components, the total engineer payload is approximately 1,872 base pairs. Staying well under the strict 4,500 base pair packaging limit of the AAV virus is mathematically critical. It guarantees high manufacturing efficiency and prevents the creation of truncated, broken genomes when ordering the vector from a commercial contract research organization. The entire molecular blueprint is fully assembled. We have a highly efficient, a precisely targeted, and easily packaged cure. The final challenge is purely physical delivery. With the AAV9 payload engineered, we must determine the exact mathematical and physiological vector to physically dose the central nervous system. The first option, intravenous systemic delivery, is highly inefficient. Because the blood-brain barrier blocks most of the virus, it requires massive viral doses, around 10 to the 14th vector genomes per kilogram of body weight. Pumping that much virus into the bloodstream creates severe risks of liver toxicity. The second option, direct stereotactic injection into the brain parenchyma, maximizes local transduction but requires highly invasive brain surgery, creating extreme physical danger, lack of scalability, and prohibitive facility costs. The optimal pathway is the third option, intrathecal delivery via a standard lumbar puncture. A lumbar puncture injects the vector into the lower spine, entirely below the termination of the spinal cord. It deposits the payload directly into the cerebrospinal fluid, completely bypassing the blood-brain barrier. Because the fluid carries the payload directly to the brain, this intrathecal route achieves potent central nervous system transduction using a viral dose 100 to 1,000 times lower than systemic intravenous routes. The procedure takes minutes, uses basic, inexpensive medical equipment, and requires no specialized neurosurgical infrastructure. If the molecular engineering is a solved equation, and the delivery method is a routine outpatient procedure, why does this cure not exist on the commercial market? We must properly categorize the therapeutic scope. For NT1 patients with a documented missing peptide, the therapy directly restores the deficit. For NT2 patients, where the specific pathophysiology is less certain, the therapy relies on brute force mass action effects to artificially elevate arousal. This ledger shows the real-world math. The exact cost to procure a medium-scale research-grade batch of our engineered AAV9 vector from a contract research organization is roughly 2,500 to 4,500 pounds. Combined with basic lumbar puncture procedure equipment, the total one-time cost of the cure is around 7,000 pounds. This graph maps that flat 7,000 pound fee against the current commercial model. A lifetime of symptomatic management with sodium oxybate and stimulant polytherapy results in a cumulative cost of 1.2 million pounds per patient. A pharmaceutical company accustomed to 1.2 million pounds in recurring revenue over the lifespan of a single patient is actively disincentivized to develop a 7,000 pound one-time intervention. Furthermore, the regulatory development cost for FDA or EMA approval typically exceeds 50 to 100 million pounds. This mathematically destroys the profit margins for a single-dose treatment in a small orphan disease population. The absence of a commercial orexin gene therapy is purely an economic phenomenon. It is a market failure, not a reflection of scientific, biological, or technical limitations. The technical framework for clinical translation is already complete, documented, and openly available. Decentralized medical networks armed with existing off-the-shelf biotechnology now have the exact blueprint required to bypass broken commercial incentives and implement the cure directly.